Yes. We can see your cursor.
Okay.
I can
Yes,
see.
sir. Yes, sir.
Yeah.
Okay. So this is just one well of the 96 well plate. In the plate, there are 96 wells in which we are talking about the one well. This one well of the 96 well plate has the antigen, which is coated. Coated in the sense that is, it is firmly held in the plate. What is this antigen? It can be a protein, it can be a polysaccharide. Now, the next step is what we do is there are many other steps which we'll be discussing in the later slides, but what are the major steps? First, we coat the antigen against which we want to see the IgG or the antibody response. Now, after the coating of the antigen, which is generally a protein or a polysaccharide, we put the primary antibodies. In vaccine samples, generally the primary antibodies is the sera samples either from the animals or from the humans. Why I'm saying animals or humans? Because when we develop the vaccine, generally the vaccine is seen first in the animals. What is the whatever the suitable animal model is there, we see the immune response in that. We inject our vaccine, and after a certain period of time we take the antibodies which are present in the sera. So we take the sera and then put it in the well, which is already coated with an antigen. Now, this antigen or the antibodies which are present in the sera are very specific for the antigen. So it goes and bind to the antigen, firmly bound. Now, the next step is what we do is after the first and second step, that is the antigen is coated, then we have the sera samples, which has the primary antibodies. Now we put the commercial antibodies, which are anti-antibody. Anti-antibody, what does this mean? That is the primary antibodies which are coated to the antigen. The secondary antibodies, you can see that these are secondary antibodies. These are very specific against the primary antibody. What they do is they go and bind to the primary antibodies very firmly. On the other hand, the secondary antibody is coated, not coated, is bound to an enzyme, which in this ELISA technique is the peroxidase enzyme. So now we have a complex. In the third part, if you see, we have a complex wherein we have the antigen, then we have the sera from the mice or the humans, animals or the humans, whatever sample we are taking. Then we have the secondary antibodies firmly bound to the primary antibodies and then attached to the enzyme. After this complex is firmly there in the well, and remember, I'm talking about one well, and this is how all the wells behave in 96 well plate. Then after this, we put the substrate. What is the substrate? Generally, it is trimethyl benzidine for the which is specific for the enzyme. The moment we put the substrate, there is a color change, and that color change we estimate. So now, if you see, if I just generally talk about the principle, the antigen is bound to that antigen is the primary antibody bound to that primary antibody, the secondary antibody is bound, which is linked to the enzyme, and then the substrate is put, which gives you a color change, which means the more is the primary antibody, the higher are the chances that the secondary antibody will be bound to the primary antibody, and more will be the color change because the enzyme will be more. Suppose if you have in any sample less of primary antibodies or less of vaccine response, it will what it will do is that less of secondary antibodies will be bound to it, and then there will be less of color change. So in simple terms, you can say that the higher the immune response, the more is the color change we can see. And why we are calling this as indirect ELISA? Because we are estimating the primary antibodies or the immune response using secondary antibodies. We are not directly estimating. Directly in the sense, had we be had we would be having the primary antibodies linked to the enzyme, then we would have measured the direct through direct ELISA. Since here it is primary antibody which is not linked to any of the enzymes, and the secondary antibody is linked to the enzyme. So that is why we are estimating the immune response using indirect ELISA. So this is the kind of basic principles I'll be moving ahead with more details about ELISA, but then just keep it in your mind that these are the primary and basic steps. Anywhere, if you have any questions, you are most than welcome to put up your questions. Thank you. Okay, now going on to the next slide. Apart from the indirect ELISA, what are the other types of ELISAs which are being done? So we have direct ELISA, which I told that instead of putting the secondary antibodies, if you see in the direct ELISA, what is done is we take the first step, which is the antigen coating to the well. Now we put the primary antibody, which is generally the sera samples from either from the animals or from the humans. But then in this case, the primary antibody is linked to an enzyme which directly gives the color change once we put the substrate, and then we estimate. This is kind of little difficult compared to the indirect ELISA, which is little simple, because in primary antibodies, it is difficult to link it with the enzyme for the immune response, I'm telling you. Indirect ELISA, I already told you. Then you have the sandwich ELISA, where what you do is that you have two antibodies, that is the capture and the detection antibody. You code and remember, the antibodies are basically the protein. So you code the well with the capture antibody, then you have the antigen, and then you put the other antibodies, and then you have the secondary antibodies. This is sandwich ELISA. Competitive ELISA is that you have the competition between two things. Now, what it means is it's a format where the sample antigen competes with a labeled antigen for the binding to the capture antibody. Competitive ELISA is generally done to estimate the concentration of the antigen. How does this differ from the indirect ELISA is? In indirect ELISA, we measure the immune response, that is the antibodies. In competitive ELISA, generally we measure the antigen concentration. If we are not sure, for example, if we are running a batch of, um, any of the fermentation batches we are doing for the development of the antigens, which we want to take it ahead for the injection in the mice or any of the other animal models, and we want to specify and we want to quantify the antigen concentration per ml or maybe higher than that in terms of the yields. We do the competitive ELISAs uh, with regard to the standards, and then we estimate our antigen. So it is a kind of, uh, basically it is also for the identification and quantification of the antigen. So these are the different types of ELISAs which are being done for the vaccine development. Some of them are used for the immunogenicity, some of them are used for the antigen estimation. Any questions? Until now, I have discussed two slides, or maybe I'm too boring in terms of explaining you the things. Let me know if you have any questions. Uh, I'll be more than happy to answer it. Let us wait for 2 or 3 seconds. If you have any questions, anybody can raise the hand.
So please do not hesitate, even if it is a very basic question. So please do not hesitate to ask.
I know it's on Sunday, but then still, I'm quite sure I'll not ruin your Sunday.
Yeah.
Okay.
So
Okay,
I
let's
guess,
move ahead.
yeah.
Okay.
Yes, 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 yeah, sure.
Now we have the key components of ELISAs. What are they saying? for the ELISA, certain things are required in terms of the reagents, in terms of the samples, in terms of the equipments. First, we need is the ELISA plate. Generally, the ELISA plate, which is the type of ELISA plate which is required in the immunogenicity assessment, is a polystyrene ELISA plate. You must be having a question that why do we use the polystyrene ELISA plate? Polystyrene ELISA plate, it is very much suitable for the immobilization, for the coating of the antigen. And I hope you remember in the very first slide, I told you that the antigen can be a protein or a polysaccharide. However, it polystyrene coated wells or polystyrene coated 96 well plate is more suitable for the protein antigen coating. If the antigen is a polysaccharide, it is not firmly held on the polystyrene well. So what we do is we have a mixture in which we take the protein, which is a kind of non-specific protein, which is not similar to the antigen. Here in this case, the antigen will be polysaccharide. We mix it with a protein which will not be having, which will not be giving any kind of readings or any kind of hindrance in the ELISA. We mix it and then we coat it. So what it does is that the protein goes and bind to the well. Side by side, it is also bound to the polysaccharide. So we, in a way, we estimate the IgGs which are present in the primary as primary antibodies in the sera, that is being estimated against the polysaccharide. Why do we use protein if the antigen is a polysaccharide? Because polysaccharide does not go and bind well to the polystyrene plates. Next is the target antigen. As I told you that the target antigen can be a protein or a polysaccharide. Will not be going into so much details because you already have an idea that it can be a protein or a polysaccharide. Okay, now the primary antibody, I told you that generally the primary antibody are the sera samples. Just a second, there is uh, okay. So primary antibodies are generally the antibodies which are found in the sera samples of the animal models. If we are testing the animal models for the immunogenicity or for the immune response. Other than that, if we have the human samples which are there for the clinical trials, there in the sera samples of the humans, we have the primary antibodies. Secondary antibodies are generally the anti-antibody which is against the antibody of against the primary antibody, and this is generally commercially available. Yes, I heard a sound. I think there was a question. No.
Is there any query? Please unmute yourself if you have any query.
Okay,
Yeah.
so the secondary antibody is the anti-antibody. For example, if you are looking the immune response in mouse. So the secondary antibody in that case will be anti-mouse, either raised in goat or raised in rabbit. So how do you pronounce or how do you designate it as goat anti-mouse? Goat means it is raised in the goat against the mice antibodies, which are the primary antibodies. Okay. Now the blocking or assay buffers. We have blocking or assay buffers because we require these kind of buffers while we are moving ahead or doing the whole procedure of ELISAs. The composition I'll be telling you, generally the blocking buffer is phosphate buffer saline, which is with a protein, non-specific protein, so that it binds to the non-specific binding sites and reduce the background noise and does not give us false positive results. Then we have the substrate solution. Already I told you that the substrate solution, this substrate is for the enzyme which is linked to the secondary antibody, which is here in this case is the peroxidase enzyme. The substrate solution is basically trimethyl benzidine. For NPP, which is nitrophenyl phosphate, it is alkaline phosphatase. So the substrate which is generally put, or not generally, it is put in the end as a stop kind of solution to see the color response, to see the color which has been developed. The TMB is for the peroxidase enzyme specific, and for PNPP, which is nitrophenyl phosphate, for alkaline phosphatase. Then we have watch buffers, because it is not like that we have to do the ELISAs with the coating of the antigen, primary and secondary antibodies. We have to wash it also, because whatever antigen or the primary antibody or the secondary antibody which is bound to the complex, it goes and bind, and then the rest of the amount of the antigen or the primary or secondary antibodies which are not required, which are unbound, they have to be washed, and they are done with the wash buffer. Okay. Now, coming on to the next slide, we have the advantages and disadvantages of ELISAs. Obviously, any of the technique or any of the tool has some advantages, some disadvantages, but then because of the because they can be used for the antibody estimation, they are being used, keeping in mind both the advantages as well as the disadvantages. In terms of the advantages, we have the higher sensitivity. The reason is that the use of the labeled secondary antibodies allows for signal amplification, making indirect ELISA more sensitive than direct ELISA. Why more sensitive than direct ELISA? Because when we put in the very first slide, I told you that we put the antigen, coating the antigen, then we put the primary antibodies, then we put the secondary antibodies. So in that complex, the secondary antibodies, they are specific to the primary antibodies. So it is having a higher sensitivity. That is why the higher sensitivity is one of the advantages of ELISA. The other advantage is the flexibility, which says that the primary antibodies may be different for different host species, because it may be different for hib, it may Haemophilus influenza type B, it may be different for Neisseria meningitis. But then if you are having an animal model which is mice, the secondary antibodies can detect multiple primary antibodies. I'm not saying that you are detecting two immune responses, or the immune responses of two different diseases, infectious diseases at one time. No, what I am saying is that if you are doing the ELISA for Haemophilus influenza type B, and if you are using secondary antibody labeled as A, that same secondary antibody labeled as A, and that too for mice, which is mm, raised in goat, but it is very specific to the mouse antibodies. It can, the same single secondary antibody can be used for the other infectious disease in another ELISA method, as it will be similar for the mouse primary antibodies. So that is why it is having flexibility. And then we have the broad application range, which says that this format is suitable for detecting specific antibodies in various biological fluids. It can be applied in diagnostic tests in terms of identifying the antigen, in terms of the vaccine development, that is the immune response, and obviously various other applications, either in immune responses, or in diagnostics, or in various other fields. Now, coming on to the disadvantages, there are chances that it can cause cross reactivity. Why? Because it can give false positive results, because secondary antibody, although it goes and bind to the primary antibody, but then it can recognize so many other primary antibodies. So it can go and bind to too many different kind of primary antibodies. But there are ways and means how to tackle it, and then we do it in the ELISA to make sure that it is not binding to any other any other primary antibodies which are non-specific to that infectious disease in terms of having different kind of samples. We rule out that to rule out the false positive results. Complex protocol. I would say complex and laborious protocol. Indirect ELISA, it requires multiple incubation steps, washing steps, and then high precision. Good pipetting, technique is good pipetting hands are required, and it requires time. It is generally done in 2 or 3 days. Sometimes people do in one day, but I would never recommend doing it in one day. Generally, it is from 2 or 3 days. Okay. Uh, Coming on to the applications and equipments of ELISA, it can be used in diagnostics for clinical samples to identify specific antibodies such as HIV or allergy testing, where sensitivity and specificity are critical. You have to be very specific and highly sensitive that the number of antibodies which are required for the infection are there in the sera sample of the patient. There you can apply ELISA as a technique for the kind of right diagnostics so that right treatment or intervention can be given against that disease. For the vaccine development, it monitors the immune response by detecting antibodies against specific antigens. Antigens can be proteins or can be polysaccharides. It gives you the IgG, whatever you are estimating, either IgG or IgM in terms of its units, which is generally ELISA units per ml. So it gives you the correct number. It can it can quantify. It is not a qualitative test. It can quantify the antibodies. Immune research, this is quite varied. The field assesses immune responses in various studies, such as those involving autoimmune diseases, infectious diseases, or therapeutic antibody development. So it can be used in the therapeutics, it can be used in the infectious diseases, autoimmune diseases. ELISA is applicable to so many different kind of, fields. Quality control in biotech, obviously you have to have a quality control in place so that there are all the checks and balances once you are doing these assays. It ensures that the presence and concentration of these specific antibodies or the proteins in the biological fluids, cell culture, supernatants, or purified protein preparations are up to the mark. What is their concentration? Or even if you are identifying the antigen, what is the concentration of the antigen? If you are estimating the antibodies, what is the concentration of the antibodies? So it is a kind of quality check also. In terms of the equipments, what is required? Precision pipettes. Pipettes are very important. Here, I can say pipettes, pipetting and good pipettes, which are well calibrated, are important for all the molecular biology techniques, be it PCR, be it ELISA, or in any other, molecular biology techniques, because if there is an error, it will give you results which will not be true, it will be false, and can lead you to disastrous consequences if you are developing a vaccines or even in the therapeutics. So pipettes should be precise, well calibrated, and then it can be used. ELISA plates, generally, I told you that the ELISA plates which are used in the immune response assessment are the polystyrene coated plates. Plate washer, obviously, you can do hand washing also, but then generally it is not recommended. What is recommended is an automated plate washer, which is there for the 96 well plate. The reason is that after washing, the residual volume of the or the leftover volume in the well should be very, very less. It is generally between 1 to 5 microliter. So which can be achieved with good plate washers. If you are doing hand washing, it can give you more than 5 or 10 microliters of residual volume in the plate or in the wells per well, and it can give you different results, which will be kind of which will be giving you errors. Then you have incubator. If you want to coat the antigen quickly, you just put it at 37 degrees centigrade. Otherwise, generally, most of the ELISAs are done at room temperature, which is 23 plus minus 2 degrees centigrade. Then you have the ELISA reader, or you can say microplate readers, various companies, various standard manufacturers. They give the ELISA readers which have different wavelengths. Generally, the indirect ELISA, which we do for the immune response assessment, it is the samples are read at 450 nanometers, which is the yellow color. If we are putting the stop solution, which is the H2SO4, and if we are reading at the substrate, the step which where we add the substrate and then we get the blue color, it is at the 630 nanometers. So various ELISA readers have various kind of filters fitted, for which you can have the readings or the optical densities, ODs, attained, and then you plot it for the standard curve and get the IgGs. Hope I am not so quick in terms of explaining. Just let me know, please. I can be a little slower also. Dr. Isha, is it fine?
Yes, it is fine. And even I have a query, like
Yes.
Dr. Sharma, like when some like we have faced different challenges during the last pandemic when come. So when certain new pathogen come, so what are the challenges scientists face while designing the ELISA for the for the new pathogens, detection of the new pathogen?
For the detection of the new pathogens, Dr. Isha, one of the biggest problem is that you don't know the antigen
Mm-hmm.
which is giving you the disease. In some of the, kind of diseases for which the ELISAs are established, you have the well-established WHO certified antigens available, which are
Okay.
quoted
Mm-hmm.
in the required quantity. For
Okay.
COVID or for any other emerging pathogens, the
Mm-hmm.
difficulty is that you don't have the antigen. The second is you don't have the positive controls, not positive controls of the study, but then the
Yes.
positive control which is put in the plate against which is a kind of standard curve.
Yes.
So
Mm-hmm.
that standard curve is difficult to make for the emerging pathogens. Then you
Mm-hmm.
have the optimization, because
Okay.
results vary from lab to lab, person to person. If
Mm-hmm.
there is a disease, for example, Neisseria meningitis, for which that the vaccines are available, commercially available in the market.
Yes.
For these
Mm-hmm.
kind of diseases, already
Mm-hmm.
the ELISA methods are well established. For new
Okay.
diseases, it is
Mm-hmm.
little difficult. It will take time, and you have to be
Mm-hmm.
double sure that what kind of results you are getting.
Okay. Okay. Mm-hmm.
Yeah,
Understood. Yeah. Thank you so much. Thank you.
thank you. Now, why I'm repeating indirect ELISA again and again, so that you absorb this very well, the whole process. This is actually quite interesting. I'm not saying because I have done ELISA. I'm saying this because ELISA per se as a technique is extremely important and very interesting. I hope you people move ahead in your career and do ELISAs for diagnostics or for the vaccines. I'm sure you will be enjoying it. So here it gives you a detailed view how the antigen coating is done. What we do is we take the concentration of the antigen. This can be 3 microgram per ml. It can be 1 microgram per ml, the concentration of the antigen, or it can be 5 microgram per ml, which is taken to be coated in the 96 well plate. And remember this, if this is, for example, 3 microgram per ml, it will not be that you are putting that whole antigen 3 microgram in one well. It will be only 100 microliter of this volume. It will be going for the coating antigen, so that this coating antigen will be somewhere in nanogram. for Neisseria meningitis or for this Haemophilus influenza type B vaccine, I can tell you it is generally 500 nanograms, sometimes 200 or 300 nanograms. That needs to be optimized. But then this is the stock concentration of the coating antigen, and then 100 microliter per well you coat it. So it comes in the nanogram range, and then you uh, just cover the plate with an adhesive plastic, or you have a plate cover also. You incubate at 4 degrees centigrade overnight. If you want to do the next step, which is the blocking step, for example, after 2 or 3 hours or 4 hours, you can put this coated antigen coated plate at 37 degree centigrade, so that your reaction is little faster compared to 4 degrees centigrade. You all may be knowing that at lower temperatures, the reactions they become slower. So it is uh, keeping the plate at 4 degree or at 37 degree. It depends how you want to develop your ELISA. It depends how you are taking your ELISA ahead in terms of the further steps. Because it's a laborious, test. So people generally keep at 4 degrees centigrade, and some of the researchers or some of the good scientists, they say that at 4 degrees centigrade, if you leave it for the overnight, if you leave it at 12 hours, the coating of the antigen is much better compared to what you do at 37 for 3 hours. But this is, I'm not going into the debate, but then you can have different temperature range for the coating, and then you leave it. For example, if you are taking 4 degrees centigrade, you put it in the refrigerator, which is 2 to 8 degrees centigrade, generally maintaining a temperature of 4 degrees centigrade. You keep it overnight. Next day, you take out the plate, put it to the room temperature. And remember, while you are doing the ELISAs, never ever do this. That you just take out the plate at 4 degrees centigrade if it was kept overnight, and start doing your ELISA. At least bring that plate to the room temperature, and then you do the next steps. Now, for the blocking, what you do is you have antigen coating buffer, which is generally phosphate buffer saline. Then you have a protein, either BSA in that PBS, and you have a detergent also. You put it, you mix it, make the blocking buffer or the assay buffer. Why I'm saying blocking and assay? Because blocking buffer and assay buffer are the same. Assay buffer is used in the further steps of ELISAs in terms of the twofold serial dilutions. So blocking and assay buffers are generally the same. Now, what you do is you block it with generally 200 microliter of blocking buffer per well to block the non-specific protein binding sites. And remember, for this blocking buffer, you have this, non-specific protein, either BSA. Sometimes I can tell you, for Neisseria meningitis, we have used fetal bovine serum, uh, this protein, and then you cover the plate and put it at 37. Here they have written 37. We used to keep it for 2 hours for at room temperature. So it is, see, I'm telling you, it is different as per the lab. It is the basic steps of ELISAs will be the same. However, there may be change in the temperature. For example, 4 degree overnight. This can be for the blocking, it can be at 37. For us, when we develop the ELISAs in-house, we optimize, and then we did so many runs just to see if there is any difference at 37 or room temperature. We found that more or less the results are the same. So we did the blocking. We started doing the blocking step at room temperature. Okay. Now, the third, see, this was first and second step. Now you have third and the fourth step. In the third step, you do the antibody incubation. Remember, once you coat the antigen, you do the blocking, and then you do the washing, you put the antibodies. What are the antibodies here? These are the primary antibodies, either from the sera of the mice or from the human samples. You do some of the dilutions, pre-dilutions, then you do twofold dilutions in the 96 well plate. I'll be explaining in the later slides how to do it. And then you put the primary antibodies in the 96 well plate, which are already coated and blocked. So these primary antibodies go and bind to the antigen against which you want to estimate the immune response. Then you do the washing again after putting the primary antibodies. Then you put the secondary antibodies, which are anti-antibodies. These are commercially available. You coat them, and then although they have written at each and every step at 37 degrees centigrade, but then I can assure you that these steps can be done at room temperature also, because we have done it. We established that. Now then again, washing is done, and then for the signal detection, what they do is they put the TMB. What will TMB do? TMB is a substrate which is for the enzyme which is attached to the secondary antibody. The moment you add the substrate, it will give you a color change. Generally, TMB color is blue color. It is red at 630 nanometers. Read the if you want to read the TMB, you can read at 630 nanometers. Otherwise, if you want to stop it, you stop it with 2 molar H2SO4. And then the color from blue, which is the substrate color, which is given by the adding of the substrate, it changes to yellow. This yellow is read at an absorbance of 450 nanometers. So now you have various samples. For example, you have different kind of samples of different doses. So you will be having different ODs present at 450 nanometers. So these are basically the steps of ELISAs. Now, this is quite important. You have to understand this. And I would say, please ask any of the questions if you are having, because this is very, very important once you are establishing your essay for the vaccine. Here in this case, I have taken hemo. I'm so sorry. It is actually written Haemophilus in influenza. Sorry for that. It is actually Haemophilus influenza type B, which is Hib vaccine ELISA for preclinical samples. Preclinical in the sense that it is in the animals. So starting from the first point, what are the animal species? Generally mice. Generally mice. For Hib also it is mice. For Neisseria meningitis, although I'm not talking about Neisseria meningitis, ELISA, but for Neisseria meningitis, the known or the suitable animal models in which you see the immune response for your vaccine is mice. Coating antigen here is the polysaccharide. Uh, uh, I hope you remember I was talking either in the first or the second slide that your coating antigen can be a protein or a polysaccharide. Here, in this case, it is a polysaccharide. So it can be a problem because the 96 well plate which you are using is a polystyrene coated polystyrene 96 well plate, which is not very suitable for the coating of the polysaccharide antigen. So what you do is, as I told you before also, you mix the polysaccharide with a protein which will not give you any kind of hindrance or results with your ELISA. You just mix it so that protein goes and bind to the well, and simultaneously, side by side, it is also bound to the polysaccharide. So in a way, you are basically estimating your immune response against the polysaccharide. Here in this case for Hib, also it is the polyribosyl ribitol phosphate, which is the name of the polysaccharide, which is the antigen, which is a kind of virulence factor for Hib, for which majority of the vaccines are being made, and they are quite successful. Primary antibodies, where are they? In mice sera. Secondary antibodies, generally commercial antibody against primary antibody, and these are enzyme linked, and these are basically commercially available. Sera, how how the sera is very important to you. Why? Because this has to be collected for the IgG estimation. And you should note that why I'm saying IgG all the time or most of the times in this presentation, because in Hib, basically, we estimate IgGs, immunoglobulin G, because these are the long-lasting antibodies, persistent antibodies, which are responsible for giving you protection against Haemophilus influenza type B infection. That is why if your vaccine is giving high IgG concentration, it means your vaccine will be working. I'm not saying that it will work. It means that it may be working. There are other tests also which are kind of confirmatory test apart from ELISA, by which you also confirm that this vaccine is working. And then for the IgGs, these are the persistent antibodies. That is why you identify IgG anti-PRP antibodies. What does this mean? Basically, IgG against anti-PRP. Anti-PRP is basically the polysaccharide, polyribosyl ribitol phosphate, which is the polysaccharide, which is the antigen. Against that antigen, when you injected in the mice, they gave you the antibodies. What are these antibodies? These are basically the persistent antibodies. What are persistent antibodies? These are basically IgGs, and that you estimate using an indirect ELISA or using a commercial antibody in terms of higher ODs or in terms of the ELISA units per ml. This is how you do. Now, the collection points, before going on to 6, 7, 8, and 9, let us uh, go through the HIV vaccine dosing schedule. For example, you have a vaccine which you want to test, and for which you want to estimate that what are the IgGs which will be generated in a mice or kind of, mouse model. How to give it, when to give it, how much should be given. For this, you have to have a dosing schedule. If you see at the bottom left, you have the first dose. At this first dose, you just take one animal, which is mouse hair. You give the first dose, you have your vaccine, which is properly formulated. Don't confuse yourself with the formulations. I'm not going into the details of the formulation. Formulations basically make the vaccine antigen stable, but then the basic component of any formulation is the vaccine antigen. So you give the first dose at day 0 in the mice, and then before giving the dose, you collect the blood at day 0, just to make sure that there should not be, if you are giving the first dose, before that, if you are collecting the blood, it can be very conveniently assumed that it will not be having any immune response, because you are just giving the antigen to that animal. Okay, first dose. Second dose is at day 14. You, before giving the injection at day 14, this is the second dose in the same animal. Just consider one animal. For that same one animal, you gave first dose at day 0, and before giving the dose, you took the blood, and then at the second dose, you have which is at the day 14, you take out the blood before giving the second dose, collect all the blood samples, because you have to segregate the sera from these blood samples by doing centrifugation and all that. Collect it, keep it at -20. Generally, it is kept at -20. Or it can be kept at 2 to 8 also, but I would suggest -20. I think it's better. And then at day 28, once you have given two doses to the same animal, one animal, and this will be repeated to so many other animals having different doses. Don't go on that. Just consider one animal at a time. Then you euthanize that, in the sense you kind of, it's a terminal bleed. At day 28, you are not giving any dose. You do the terminal bleed. At the terminal bleed, you will be having more and more kind of more sera compared to what you got at day 14 and day 0. Obviously, the mouse was alive at that time, So you will be having less of sera samples with you, less of sera volume, sorry, not samples, less of sera volumes. At day 28, which is the terminal bleed, you will be having more of, sera volume. Now coming on to the hip vaccine study design, just just for the just as an example, I have put this, the design in which you have group one, which is the negative control. Obviously, in any of the essay, if you are doing, you want to see what are what are the estimates or what is the value for the negative control, which should be kind of baseline, very close to zero, although it is not zero, it may give you some values, but very close to the baseline. Then you have group two, which is the positive control, and the composition of negative control is phosphate buffer saline generally. However, people keep different kind of negative control, but then it is not having any antigen. It will be giving you least response, that is very close to the baseline. Then you have group 2, which is the positive control, which is the comparator vaccine. Why I'm saying this, if you have a vaccine available in the market, then you buy that or you procure that, and you have to see whether your test vaccine, which is it, group 3 or group 4, is kind of non-inferior or kind of superior to the comparator vaccine. Why I'm saying non-inferior and superior, because these two terms are very important, because once you move ahead at different steps, the regulators will be asking you that there is a comparator vaccine available in the market. How can we be so sure that your vaccine is kind of non-inferior, in the sense very close in terms of giving the concentration of the IgGs to the comparator vaccines, or it is superior, that is, it is giving IgG values more than the comparator vaccine. I would be a little conservative. Generally, what is seen is non-inferior. Superior, extremely good, but then generally what we aspire or aim for is the this non-inferiority. Okay, now come back to point number 6. Now the collection points, you may be quite little bit clear that the collection points, blood collection points will be at day 0. If you see here, day 0, then day 14 and day 28. Sorry. So 3 collection points.
Dr.
Groups in
Sharma,
these, yeah,
yeah, sorry to interrupt. There is a question from a participant, like he has raised.
yes, yes, please
Mm.
tell me.
Yeah.
I
Good
can't
morning,
see
sir.
the question. Yeah, yes, please go ahead.
So, myself, Yatin, sir, I wanted to ask the question that what are the more factors or the characteristics that make one vaccine more inferior and one vaccine more superior than the other one?
See, there are so many factors. I was thinking on that. There are so many factors which may make the vaccine non-inferior, superior, or maybe inferior to the comparator vaccine. For example, if you have a vaccine which is in the commercial market, which is like having an antigen dose of 20 microgram, okay? Commercial vaccine, and you make a vaccine of the same kind of antigen, same properties of 20 microgram, and it is not giving you the same response, there can be several reasons. If I take the example of polysaccharide conjugate vaccines, any polysaccharide conjugate vaccine which is available in the market at 20 microgram is giving you a good response compared to what your 20 microgram same polysaccharide conjugate vaccine, either for Hib or for Neisseria meningitis, is not giving you the same response, can have so many reasons. Why? Your antigen is not estimated properly. Either it is your processes are not estimated properly. What you are calculating as 20 microgram is not actually 20 microgram. It is either 15 microgram or maybe less than that. The other point is that in the conjugate vaccines, you have the antigen, which is generally a polysaccharide, conjugated to the protein. Okay, the carrier protein, which gives you the T cell response. If the conjugation in your vaccine is not properly done, it will not be a proper conjugate. So there are chances that it will give you a lesser immune response. Other things which are responsible can be free polysaccharide, free protein, which are giving you the non-inferior, inferior response. So that is why I am telling you that there can be n number of reasons which can be related to your process. There can be n number of reasons which can be related to the QA/QC check. There can be n number of reasons which can be related to the formulation of the antigen. For example, if you have 20 microgram commercial vaccine giving you good antibodies, and you have your test vaccine which is 20 microgram, properly conjugated, properly estimated that this is 20 microgram, but then it is not properly formulated, it means over a period of time that is not stable, that is degraded. Your process was good, your antigen estimation was good, it was 20 microgram, your conjugation process was good, it was properly conjugated, that is polysaccharide was conjugated to the protein very tightly and properly, but then your formulation is not good. It is not keeping the antigen stable. So even if you have extremely good QA/QC parameters for the antigen, for the conjugate, but then your formulation excipients, for example, the buffers are not properly prepared and they cannot keep your antigen stable, that will be the reason for the low inferiority. I just gave you two or three reasons. There can be n number of reasons, because vaccine development in itself is a complex process. Did I answer your
Yes, sir.
question?
Yes, sir.
Was that
Thanks,
too
sir.
elaborative or very short for you?
Sir, it was rather that I could search for more, but it was something that I could hold on to. Like I can look into it myself then. It was well explanatory.
Thank you. Thank you so much. So should I move ahead?
Yes, sir, sure.
Okay. Now for the test samples at day 0, if you see number of mice we have taken in each group is 10. So if you calculate for the 4 groups, it will be 40, obviously. At day 0, if you see how many samples you will be having at all the different time points, either day 0, 14 or 28, number of mice will be the same, 40. So the number of test samples at day 0, 14, 28 will be 40. How many samples you have to evaluate is 120. So generally, it is not like that we have 40 animals, we have to establish, so we have to estimate, IgGs in 40 samples. No, it is having a different time point. So it is like 120 different samples at different time points you have to estimate for the IgG concentration. Now the injection, see in mice, in humans, generally what is seen is anything in terms of the vaccine antigen, if it is injectable, it is intramuscular and generally 0.5 ml. Okay. In For the animals, if the injection is there, generally it is subcutaneous and 100 microliter. Why 100 microliter and 5 ml in humans and 100 microliter in animals? Because if you are giving 200 or 300 or 400 microliter, it may spill and the antigen will not be going inside properly, will not give you the immune response. See, this can be one of the reasons for a low immune response, which can be seen in the animals. Got my point? So the volume is 0.5 ml injectable in humans. For the vaccines, I'm telling you. For therapeutics, it will be different. For animals, it will be 100 microliter. So is this slide clear to all? Should I move ahead? At least I need 10 hands to be raised. Then only I'll move ahead. Or else I'll say thanks and then we'll stop the presentation.
Yeah. So Dr. Sharma, yeah.
Yeah, I think we
Floor
have
is open.
many hands
Really.
raised. Yeah, thank you. Go ahead.
Excuse me, sir.
Yes.
Uh, Good morning, sir. This is
Good
this
morning.
side, Dr. Manju. The
Yes.
nice presentation. My one
Thank you.
question is, I'm from plant science background. I just want to know, can we use this ELISA technique in drug and plant-based therapy studies for a specific diseases?
Yes.
For example, we have extract of any medicinal plants, say for any kind of diseases, for example, for neurodegenerative diseases. So can we use this technique in that case for plant-based therapy?
Yes, plant-based therapy, I think you can use, but then I'll be specific that generally in the ELISA, what you estimate is the concentration of the protein, which can be the
Right?
antigen.
There may be
So,
specific biomarker related to that particular kind of disease.
you have these biomarkers as proteins.
No, I mean to say that can we use this ELISA technique in drug and plant-based therapy studies for a specific disease?
Yes, I would say it's a yes from my side, but then with a clause
Okay, right.
that in generally in ELISA, what you estimate is the protein, either in the form of the
Right.
antibodies, which is, plants don't have the this immune responses. Okay, but then if you want
Okay,
to
right.
identify or evaluate, estimate proteins, then for sure ELISA can be used.
Okay, okay, sir.
For
Right.
polysaccharides also ELISA can be used. But for other these compounds, I'm not sure. Plants may be having polyphenols, which are these, pigments. These are
Yeah,
organic
polyphenol are
compounds.
there in plants.
Yeah.
Yeah.
So these are basically the polyphenols are basically, I think, organic molecules. I'm not sure on that. But then I'm not sure whether polyphenols can be estimated or, but then for sure, if you are developing any therapeutic molecule, which is kind of a protein molecule against any kind of disease, as you told, neurodegenerative disease, that can be
Neurodegenerative.
used. Yes, that can be used.
Okay. Okay, sir. Okay. Thank you.
Thank you. Other question?
Uh, is there
Yes.
any other question? No, no. I think we can move ahead.
Ma'am, there is one question from Gitanjali, student, one of the students.
Okay,
Okay.
okay.
There were two, three hands raised, na?
Acha, okay,
Yes.
So
okay.
one
Sorry,
was from
sorry.
her.
Yeah. Hi, Gitanjali. You can unmute yourself and ask the question. Uh,
Yes.
Gitanjali,
Yes, Gitanjali, go ahead, please unmute yourself and put up your question.
okay. Or what you can do if there is problem in mic, you can also write it down on the chat box. Okay. Okay, maybe like we can entertain her query later and
anyone else, beta?
anyone else?
See, I'll be giving my email ID also
Yeah.
to Isha, Dr. Isha. And any question, if you have at any point of time, just feel free to write to me or to Dr. Isha. We'll be
Yeah.
more than happy to answer your queries and your questions.
Yes, yes, sure.
Any other questions from any of the students?
I think not for now, Dr. Sharma.
Okay.
Yeah.
Now, this slide is again very important in terms of the HIB. I'm sorry, I just copy paste that Haemophilus in influenza type B. It is again there on the presentation. Sorry for that. Although I did a kind of recheck, uh, I think I should have put on the AI. Okay, okay. So the what we see is now, if you see here, this was my first slide in terms of the diagram or the picture. If you see this specific ELISA for this specific presentation is for Haemophilus influenza type B. So what is the antigen here? We talked about the antigen in general in the on the first slide. In the first slide, here the antigen, now it will be polyribosyl ribitol phosphate, which is a polysaccharide. It will be coated on the well. But you remember that polysaccharide generally it is not, very kind of, evenly coated, or it is poorly coated on the well. What we do is we mix it with a protein. When we mix it with a protein, it is coated well. It goes and binds firmly to the well. Now we put the Neisseria, if we have the preclinical samples, which in our case will be a mouse. So we'll be having a Neisseria, that is the primary antibody. It will go and with this primary antibody is very specific to the antigen, because this is the antigen for against which we are looking for the immune response. And this antigen has been injected subcutaneously in the animals. So the primary antibodies will be extremely specific for the antigen. It will go and bind to the antigen tightly and firmly. Go to the next step, the antigen bound, primary antibody is bound. We put the commercial antibodies here. In this case, it can be goat, raised in goat, anti-mouse, which we can also call as anti-antibody. Why anti-antibody? Because this secondary antibody is against the primary antibody. That is why we are calling this as anti-antibody. And simultaneously, the secondary antibody is bound to an enzyme. And once we have this complex in place, firmly and tightly bound, we go ahead with the next step. We put the TMB, trimethyl benzidine. Once it is there, it gives you a color which is blue, And if you want to read the TMB color, it is red at 630 nanometers. If you want to go ahead in terms of the stop solution, which is generally 2 molar H2SO4, you put it in the well, and that gives you a yellow color. And remember, all these steps, these are the major steps. All these steps are having wash steps in between, so that extra unbound primary, secondary antibodies are gone, not giving you, false results. Go at the top, it shows that the ELISA plates are coated with 1 microgram per ml PRP antigen in phosphate buffer saline. I told you that for the 96 well plate, it will be 1 microgram per ml. So you are coating 100 microliter, so it will be having 100 nanograms. For coating, it is not 1 microgram. 1 microgram per ml is the stock solution. You are just coating 100 microliter per well. It will be having 100 nanogram if the stock concentration is 1 microgram per ml. Okay. You have the serum samples or the sera samples pre-diluted. Pre-diluted, what does this mean? Pre-dilution is generally done outside the 96 well plate, so that you have. And by the way, I'm not going into the pre-dilutions because it's a little bit of, I'm not saying that you won't be able to understand it, but then we'll be requiring, at least 40, 50, or maybe 1 hour to explain the pre-dilution. So generally pre-dilutions are done outside the 96 well plate. Once you have started your ELISA, then you do the two-fold serial dilutions in the 96 well plate. That is why I have written in 96 well plate in the assay buffer. What is the composition of assay buffer? It is phosphate buffer saline plus 1% BSA plus 0.05% tween. Some people use tween 80 also, but then tween 20 is, I think, is sufficient. And then it is incubated at, just a second, hold on. It is incubated at 37 degree centigrade for 2 hours. I told you that you can also do it at room temperature. Then to detect the IgGs, anti-PRP antibodies. I already told you why this is known as IgG anti-PRP. You have biotenylated goat anti-mouse IgG added. And then it is having this secondary antibody. As I told you, it is having the peroxidase enzyme. After this, the of the well, after the steps, all the three steps in the well, which is having the antigen antibody and the secondary antibody, you add the TMB, which gives you blue color. The moment you add a secondary TMB to this complex, it gives you blue color. That can also be read at 630 nanometers, but it is not advisable. Rather than go for 450 nanometers, which is yellow color, which you will get after adding 2 molar sulfuric acid. The volume is generally 50 microliter per well. You read at 450 nanometers using an ELISA reader. Then you prepare the standard curve using CombiStat software. CombiStat software is a kind of, uh, quite a recognized software from European, I think European Quality Health, I'm sorry, EDQM, European Directorate of Quality Medicines and Health. It's a recognized software which is generally well recognized for the estimation of IgGs in EU, that is ELISA units per ml. Mm, mm, for Hib or for Neisseria meningitis or for Streptococcus pneumoniae conjugate vaccines. This is for IgG estimation. Okay, now you go on the last slide, I think. No, sorry. So now this, when if you recollect, as I told you, just a second. Okay. Uh, Go to the this slide. Remember, negative control is NC, positive control is PC, test samples is TS. Just for just to be simple in terms of the designation, I have just put this. Otherwise, you can put any of the designations you want, and as per your lab requirements. Pre-dilutions to be prepared. Three dilutions you prepare, either 1:400, 1:800, 1:1000. That is outside this 96 well plate. When you put the when you do the pre-dilutions, then you do two-fold serial dilutions. I should have written serial dilutions. My apologies for that, because it is two-fold serial dilutions in 96 well plate for negative control, positive control, and test samples. Now, this is a sample plate. Don't confuse yourself with too many plates, because you will be having ultimately 120 samples, but then the process and the steps will be the same. So take this plate as a sample plate, and this can be replicated for the other samples, keeping in mind that you have the quality control. See, Dr. Isha, you were asking that if there is a new disease, emerging disease, then you will be having problem in making the quality control. In
Okay.
For some of the diseases like infectious diseases like
Uh,
Neisseria meningitis or Haemophilus influenza type B, the quality control, which is the positive control for all the plates against which, see, this quality control will be the standard curve. Okay.
yes, yes.
Against this standard curve, you will be making or identifying all the other curves for your samples, and then you will be getting the reading, whether it is low, high, or whatever it is. So
Yes,
for
yes.
this quality control, it will be difficult for the newer diseases if they come.
Okay. Because there is no any reference or,
No reference,
no
no
reference
reference
available.
available, yes,
Yeah.
with the WHO or for example,
Mhm.
NIH or CDC. So then it
Yeah.
will be a difficulty.
Understood. Understood. Thank
Yeah,
you so
you
much.
will be having additional steps for the establishment and the optimization of the ELISAs.
Yes, yes, yes.
Okay.
Understood. Yeah.
Okay, now when you have,
And usually,
yes.
how much time it will take, like when some new pathogen came and designing all this ELISA for the, like how much time, how many months or days it take?
Dr. Isha, that is a kind of, difficult question, because if you have a good number of head count, for example, if you have 15 people in a lab,
Uh-huh.
you may be able to establish the ELISA. You may be
Okay.
able to develop the ELISA in 3 to 4 months, or maybe less than that,
Mm-hmm.
compared to if you are having a head count of 5 people.
Yes.
But generally
Okay.
that is not the case. If there is
Mm-hmm.
a severity, or there is an urgency, you have more
Mm-hmm.
and more head counts, and
Mm-hmm.
time is kind of limited. So you establish with all the checks and balances in the correct manner, you establish the essay and try to establish as soon as possible. But generally, if
Mm-hmm.
everything is in the place, you have a kind of reasonable head count in a lab, it takes around 3 to 6 months to
Months.
develop,
Okay.
and then
Okay.
to validate. You also have to validate your essay.
Regularly.
Yeah,
Yes, yes.
whether your
Mm-hmm.
essay is giving you, whether your essay is giving you the right results or not. Okay.
Mm-hmm. Yes, yes, yes. Yeah.
So you have this sample
Dr.
plate. Yes.
Dr.
Yes, go ahead.
Sharma,
Go ahead.
I just want to ask, just for the my students, can you explain the approach when there is some disease, new diseases there? So what will be the approach when we develop a vaccine for that? Just in brief.
See, the very first approach will be for the identification of the antigen. Okay. Antigen, which is surface exposed on the outer membrane generally. This can be a protein. Generally, it is a protein. But then, for example, for diseases like Haemophilus influenza type B, or for Neisseria meningitis, or for Streptococcus pneumoniae, it has capsular polysaccharide, which is the polysaccharide present on the capsule, and it is exposed and surface exposed. So you have to keep one thing in mind, that the antigen, whatever antigen you want to identify, that should be kind of surface exposed. First of all, either protein or a polysaccharide. The second thing, if it is a protein, then it should be conserved. For example, if you are, I'll give you an example of E. coli, which is, very much common for the urinary tract infections. Why we can't have a vaccine against E. coli? Because there are so many strains of E. coli which has different proteins, although the protein may be having a same common name, but then these proteins are not conserved. In the sense, they are having different sequences. Amino acid sequences are different in different strains. So even if you take a protein which may be a good antigen to be taken ahead as a vaccine, it may be effective only for example, there are 10 strains, 10 different strains. It may be effective only for 4 or 5 strains, because the protein which you have taken matches on for these 4 or 5 strains. Otherwise, for the remaining 15 strains, the protein is there, the same protein, but the amino acid sequence is different. So the immune response will be either less or extremely less, because the antibodies will not be generated in the way you want to generate. Okay. So the first is the antigen, which is surface exposed generally. It can be protein or polysaccharide, and then you have to see the conserved antigens. Sometimes what people do is they take the inner proteins also, which are present inside the membrane, which are conserved. And then they have different other antigens also mixed and put it in the formulations, and then they go ahead. So to have a broader immune response. Then the second criteria is that it is kind of conserved. हाँ तो वो हमारे strains को कर रहा है. But if you are making a vaccine for the world, global, तो उसमें तो आपको सारे strains देखने पड़ेंगे, और वहाँ conserved proteins are more suitable.
So one more question. just from the sides of the students.
Yeah,
Ki,
sure, sure.
when we develop vaccines for viruses and when we develop vaccines from for other pathogens like bacteria or plasmodium or other, so what is the difference between that? Ki, when we develop vaccines for viruses and how that is different from the vaccines when we develop for other pathogens?
See, actually, the difference is either in the technology you are using. Sometimes you have a very good pathogen, very good antigen. Doesn't matter if it is from the virus or from the bacteria. It is a difficult to express protein. Now here comes not AI or ML, but then bioinformatics. Dr. Isha, you can
Is the
also
process same, sir? Is the basic process same?
no. Actually, there are different these platforms. For example, recombinant. If I talk, see, there are different platforms. Recombinant, mRNA. Then you have for the conjugate, which is like the growth in the bioreactors, and then you do the conjugation. All three of the platforms are different. Then you have the adeno viral vector platform. You have DNA platform, which is a little different compared to mRNA platform. If I talk about mRNA, एमआरएनए डिफिकल्ट टू एक्सप्रेस प्रोटीन है, वो इतनी आसानी से एक्सप्रेस नहीं हो पाता. तो व्हाट पीपल डू इज दे पुट दी एमआरएनए ट्रांसक्रिप्ट, और वो एमआरएनए ट्रांसक्रिप्ट जो है वो बॉडी में जाके होस्ट सिस्टम, ह्यूमन होस्ट को यूज करके प्रोटीन बनाती है. अभी ये एमआरएनए कैन बी दिस टेक्निक, सॉरी दिस प्लेटफॉर्म कैन बी अप्लाइड ईदर टू वायरल डिजीजेस और टू दी बैक्टीरियल डिजीजेस ऑल्सो. तो प्लेटफॉर्म के ऊपर डिपेंड करता है. जैसे आज की डेट में इफ यू सी एंड गो ऑन द इंटरनेट एंड फाइंड आउट that what is the most most suitable and most widely used platform for either for the viral vaccines or for the bacterial vaccines, it will be recombinant. Why? Because there are so many factors. For example, cost. Recombinant platform, first of all, is an established platform. The second thing is it is easy to make a clone, do the cloning, the vector and all that. It is quite established, and it can be having two or three different antigens combined in the recombinant platform. But then if you have a very difficult to express protein, you will not use recombinant, you will use mRNA. Okay, if you have Haemophilus influenza or Neisseria meningitis, people know that polysaccharide antigen is very well a good factor for the immune response. Then they will use the conventional method, which is the fermentation of the organism, then the purification. So it depends on the platforms, and it is not like that mRNA is more suitable for bacterial. No, mRNA platform can be used either for viral or for bacterial. In the same way, recombinant can be used either for viral or for bacterial. I hope I was able to answer the question in a simple way.
Thank you, sir. Thank you.
Thank
Quite.
you.
Yes,
Thank
yes,
you.
Dr.
Thank you.
Sharma. And there is one more question from one student, Gitanjali Thakur. She is asking like which ELISA type is best for the plant pathogens. Hello, am I audible?
Yes, yes, yes. I was just thinking actually. It depends on the, no, it actually depends on the kind of antigen you are having. See,
Okay.
uh, if you want to have the concentration identified, the
Mhm.
estimation or the concentration of the antigen for against the disease for which you are developing, for example, any of the disease, neurodegenerative or any other disease, then it will be
Mhm.
for the antigen concentration and antigen estimation, it will be competitive
Mhm.
ELISA. If you want to identify the antibodies to be
Mhm.
estimated, then it will be indirect ELISA. Direct ELISA is one of the most complex ELISAs and
Mhm.
kind of used very infrequently.
Okay. Okay.
Okay, sir, I have one more question.
Yes.
This thing is in their syllabus. They have started this. When we talk about वैक्सीन डेवलपिंग वी आर वर्किंग ऑन वैक्सीन डेवलप फॉर मतलब वैक्सीन फॉर एड्स. सो अभी तक तो ये सक्सेसफुल नहीं हो पा रहा है एड्स के लिए वैक्सीन डेवलप करना. व्हाट आर द रीजंस व्हाई इट इज डिफिकल्ट टू मेक अ वैक्सीन फॉर एड्स एंड आल्सो फॉर प्लाज्मोडियम का भी हमने जो है पढ़ा है कि अभी प्लाज्मोडियम की वैक्सीन भी बनाना जो है वो थोड़ा डिफिकल्ट रहा है. डेवलप हो रही है, शायद हो जाए शॉर्ट टाइम में, कुछ आगे आने वाले सालों में. एड्स के बारे में पहले जानना चाहेंगे.
Ma'am, I'll give you a disclaimer before starting for this. See, I'm not an expert in AIDS and malaria, but certainly I can
Okay.
give you my suggestion. See, for AIDS, the biggest problem is that the antigen is not conserved. See, if you have the same antigen, but it is changing its shape from A to D, for example, A, B, C, D. If you prepare if you take the antigen which is A, you give it to the body. The moment the body encounters the virus in the form of B, the antibodies will be totally useless. Or else, if you make it in the form of B and C also, like you have two kind of antigens. See, the complexity is that you cannot have so many antigens added and then you give it to the body. The reason is that the cost will go, okay, don't, we should not talk about the cost, but then there will be complexity in the antigen or the vaccine development. So it is not very much advisable that you have 5 or 10 antigens kind of conjugated or kind of there in one formulation. So either one or two or three. But then even if you have three, and if the virus is there in the form of D, and the person, God forbid, has a chance to get the virus inside, then the antibodies which were generated for A, B, and C will not be effective for D. This is one of the primary reasons. However, there are some of the agencies like or the entities, I would say, international AIDS vaccine initiatives. I just read this on the internet that they have some immunogens. They call it as immunogens that they are developing, which they are quite hopeful for AIDS. This is one thing. Now, the second thing was malarial, I think this malaria also, they have different I don't know the stages, but I think sporozoid, merozoid, something of that sort. There are
Yes,
different
yes, yes.
stages. Yes.
Hmm.
So they target, I know a very good scientist. I don't know if you know him. Really unfortunate that he's not there with us. Dr. Deepak Gaur at ICGEB. He was
Okay.
working on malarial vaccine and he was doing very good research. So he actually had COVID and that time he left all of us. He was targeting different stages of malaria.
This
Sporozoid,
was what I
Miro.
read. Yes, sir, these
Yes,
are the things which I read in various research papers that they, the stage which they have to,
yes,
target, that
yes,
that is not, matlab, they are not getting it ki which stage should be targeted and how it could be targeted. Now, that I read
yes,
in some papers.
yes. That is a problem. Even for respiratory syncytial virus, RSV, there is one protein which they say that it's a very good protein, but the problem is that either it is in the pre-fusion state or post-fusion state. So if you make it in the if you make the antigen in the pre-fusion state and give it to the body, for example, if the person encounters RSV at the post-fusion state, then the antibodies which were being generated against the pre-fusion will not be affected for the post-fusion state. So these are, very complex challenges for different infectious diseases for vaccine development, I would say.
Thank you, sir.
Thank you.
Students, if anyone has any question, आप लोग भी पूछ सकते हैं.
I would always suggest that please ask questions. Curiosity will give you so many answers, and then you will always see that you have so many thoughts which can be converted into ideas, and then ideas into very good actions for science. Two questions. Immunoinformatics or bioinformatics is for the selection and the identification of the most suitable and the most important antigen, which will be immunogenic in nature. How do you do it? You do reverse vaccinology in some cases. For example, you know that this antigen, this protein will be immunogenic, but then you know that this will be difficult to express. Then you do reverse vaccinology in terms of the bioinformatics, and then you come to a conclusion that this protein can be made more stable, a little less difficult to express by doing some mutations, maybe some mutations, or maybe some, kind of changes. Just a second. Hold on. Yeah, so some kind of changes. So this is bioinformatics first application for the identification and for the antigen development. This is reverse vaccinology. Otherwise, the second thing is, if you don't know about the antigen, then you use the bioinformatics right from scratch. Dr. Isha, you can interrupt me anywhere because you are an expert on bioinformatics. I don't know much. For the new antigen, for the new vaccine antigen, for which you don't know, or you don't know that if there is a new disease, you have to, your boss comes and tells you that you have to give an antigen in a week. Then what you do is you start searching on bioinformatics, put all the details which are available in the public domain or with the scientific authorities and the institutions, put it in the system. You have these softwares, maybe AI, ML, or any other softwares that will give you that, okay, this is the organism, this is the structure, this is the membrane, and in this membrane, these are the proteins which are readily kind of, there, conserved also in outer membrane proteins. Otherwise, they may suggest you new antigens also. In the sense, the bioinformatics may suggest you that these are kind of neoantigens. Neoantigens is NEO, neoantigens, which are being used in immunotherapy for the cancer patients. I'm talking now about cancer vaccines. What they do is neoantigens in the sense which are suitable for a particular population or for a particular person, individual person. For example, if you if a person comes to you, he or she is of 60-65 years of age, having diabetes, having blood pressure, whatever different diseases, and you have his or her genetic makeup, then you put up all the details with all the diseases in the system, utilizing bioinformatics, AI, ML. See, I use the umbrella term AI, ML, kind of similar because I don't know much. AI, ML, bioinformatics, and then you put all the details of that patient. That system will give you a kind of an antigen which is known as neoantigen, NEO antigen, suitable for that specific patient which is suffering from that specific cancer. For example, liver cancer. That specific antigen may be suitable for that particular patient, may not be suitable for other population subset, or for other patient, because that other patient is having a kind of different genetic makeup, different kind of diseases. Even at 65, he may or she may not be having diabetes and all that. So this is kind of precision medicine or precision oncology. It is being applied in precision therapeutics also, which is the immunotherapy, which you for which you have targeted antibody, which may be bispecific, tri-specific monoclonal antibodies for a particular set of population. I hope I am able to explain you. If you want more details, we can discuss any time if you want.
Yes, sir. Thank you so much for your explanation.
Thank you. Thank you.
So there are two questions from one student, Dia Negi,
Yes.
and one is from Divya.
Yes. Yes, please go ahead, Ria
Uh,
and Divya.
so Yeah, so they have text in the chat box.
Chat box,
Um,
okay.
yeah, so like if we use indirect ELISA most of the time, what's the point of having other types? Okay. And can we use ELISA for clinical trials of vaccine and how it's used? Okay.
Okay. Answering the first question is that if you know that your indirect ELISA works well for one antigen, doesn't mean that it will be working for other vaccine antigens. So you have to have all the options on the table. For example, for the antigen identification and concentration, you use competitive ELISA. If you have a very good lab which has primary antibodies taken out from the either from the animal models or from the human samples, that is kind of conjugated to the peroxidase enzyme, then you don't need a commercial antibody. Then you will be going with the indirect ELISA. So it is the kind of what is more suitable, what is kind of available, what kind of infrastructure you are having, and what is the most suitable ELISA technique you will be having for this disease. So there are so many other factors. The second was, I think, what was the second question? I just forgot.
Uh, it is like, can we use ELISA for clinical trials of vaccine?
Yes,
And how it is
yes,
used.
yes.
Yeah.
See, specifically, if you go and search for Neisseria meningitis, for which the vaccines are commercially available, for example, GSK, sorry, Merck and Sanofi. For the approval of these Neisseria meningitis vaccines, the gold standard is the serum bactericidal assay, which is the functional antibody assay. How it is different from ELISA? Just one differentiation. ELISA gives you the total antibody concentration. What are the antibodies which are estimated? Persistent antibodies, as I told you in the presentation, are IgGs. Now, the ELISAs which you do, they give you the total IgG concentration, which is, for example, if it gives you, for example, if it tells you that there are in the by doing the ELISAs in any of the human samples, you find out that total IgG concentration was 100. For example, 100 IgGs are present. It means that out of these 100 IgGs, not all the IgGs will be functional or will be protective for the disease, will not give you protection. Maybe 60-70 will be having the will be functional in nature and will be giving you the protection. So to find out that how many number of antibodies are functional, we do serum bactericidal assay, which is the gold standard for the approval of Neisseria meningitis. Now your point is, can we use ELISA for clinical samples? Yes, because I'm saying ELISA is a laborious procedure or a technique, but compared to SBA, it is less laborious. SBA, on the other hand, is extremely laborious. So sometimes if you regulators ask, see, for phase 3 clinical trials, you have thousands and thousands and thousands of human subjects. So it is very difficult to do SBAs for all these samples. There, the regulators ask that, okay, you have done SBAs, for example, for 1,000 subjects. For the remaining 3 or 4,000 human subjects, you do the ELISAs. Because we have seen that in these 1,000 human samples, most of them, like 7 or 800 human samples, are having protective antibodies, which means that out of the total antibodies, which are 100, as estimated by ELISA, 60 or 70 antibodies are protective. Then the regulators say, okay, we generally approve on the basis of serum bactericidal assay because we want to see the functional antibody titer. But then now here you can use ELISA. So there you have a kind of liberty, and then you use clinic in the clinical samples, the ELISA technique also. Got my point? Hope I think it's clear.
Yes, sir. Yes, Dr.
I
Sarma.
hope so.
Yeah.
Yes. So
Thank you,
you
sir.
can use it, but generally, regulators say that you should be having one or two techniques simultaneously to prove that the vaccine is having the potency or kind of superior or non-inferior. But sometimes, yes, in clinical trials, ELISA can work.
Yes, Dr. Saman, I think we can also move ahead and you have well explained.
See, before moving ahead, I should give a compliment to all the students that when I was there in my post graduation, I never had these kind of questions.
Yes, yes, yes. They are very curious
Seriously,
learners. Yes, very
seriously.
nice questions. Yeah.
The question which just now I think Ria asked that these ELISAs can be used for the clinical trials. This is a very important question, which is sometimes there is a bottleneck for the approval of the vaccines because of the time problem, time constraints. Then the regulators tell, okay, then give us the data for the from the ELISAs, and then we can move ahead. And even for the there was a question, all the questions were very important. When I was in my post graduation, I never had these questions. I mean, I was not sure where to go.
Yes.
Thank you so much. Yes, so any other questions, please let me know, or else I can move to the other slide.
Uh, I think we can move ahead, Dr. Sharma. Yeah.
Okay, so before
Please.
moving on
Yeah.
to the last slide, just remember that the ELISAs, they for the ELISAs, you must have heard about linear uh, uh, a linear equation, y is equal to mx plus c. In ELISAs, we don't get the linear equation perfect kind of fit for these biological assays. For biological assays, the curve which we get is a sigmoidal curve. Sigmoidal in the sense S-shaped.
Yes.
As we have S in standard, the same
Mm.
S-shaped. If you see, this is S. The same curve we are getting here, which is kind of plateau at the end. Sorry,
Mm.
plateau at the end, then going up, log increase, and then plateau at the topmost. What we in ELISA, in the indirect ELISA for the vaccine antigen, we estimate is this part. Can you see my cursor? This is very important.
Yes, we can see your cursor, Dr. Sharma.
Yeah, so
Yeah.
this
Mhm.
this part we identify, and this red circle basically we identify the potency
Mm.
of any vaccine at this point. Okay, so always remember in ELISAs, we or for the biological assays, or sometimes people say bioassay, we have the sigmoidal S-shaped curve, and so we don't estimate this lower part, the uppermost part, but then the part which is in the between, which is in between. Okay, now I move to my last slide, which is like giving you the summary of why we do, what we require, how we are estimating. The summary is what we measure actually. We measure through ELISA the IgG antibodies. And please keep this in mind that why I'm saying IgG antibodies, because IgG antibodies are persistent antibodies, and we are measuring IgG antibodies for Hib. We can measure IgA or kind of mucosal or IgM also through ELISA. Here in this case, where the example is Haemophilus influenza type B, we have measured IgG antibodies. How we have measured that? We have coating, then we have primary, we have secondary, and then we put the TMB, and then we stop it with the H2SO4. We get yellow colored, there at which can be measured at 450 nanometers. What we required, a standard curve. If you go back and if you see that this QC, sorry, my colors, okay, this QC, if you see, is giving you the standard curve. What we require, a standard curve, which is a reference standard with the known concentration of antibodies. Antibodies in the sense, you know that this sample is having this much of antibodies. Then also, then only you can estimate and identify, sorry, not identify, but estimate and evaluate how many antibodies are there in the test samples, because you have to have a reference first. So that is why it is difficult for the new diseases which are kind of emerging or re-emerging to have the ELISA established at one go for such kind of diseases. So standard curve has to be there. How the IgG response is measured, I told you that we don't follow y is equal to mx plus c, which is the linear equation. Rather than we follow a four parametric logistic curve, which is known as 4PL model. Biological responses aren't linear. They are not linear. They are S-shaped. If you see here also, just a second, hold on. If you see here also, you see that this is the lowest part, which is kind of plateaued, not showing any increase. This is the increase. This is the again plateau. And these are A, B, C, D. I have sent the presentation to Dr. Isha. If you want, you can have this presentation. And if you have any other questions also, you're most welcome. So these A, B, C, D is A is basically the lowest part, which is known as the minimum asymptote. B is the slope. C is